Hello, everybody. Welcome again to another HIAAD seminar called Bridge, where we're going to see Dr. Chroni, Angeliki Chroni. She's a group leader, research director at the Institute of Biosciences and Applications in Demokritos Research Center in Athens. And my colleague Panos Alexopoulos could not make it today. However, we're going to have a very interesting meeting because Dr. Chroni has a lot of nice, interesting related to Alzheimer's disease. And without further delays, I would like to introduce Dr. Chroni as a very, very well-known researcher in the field of Alzheimer's disease and APOE. So, Angelika, the floor is yours.
Thank you very much, Yannis. Thank you for the invitation. I would like to thank you and the organizing committee for, you organized this very nice seminar series, and it's an honor to give a talk today. Let me share my screen. Is it okay now?
Just put full screen.
Oh, no. It should be okay now, right?
Yes, that's great. Thank you. Thank you.
Okay, okay, great. The title of my presentation is Apolipoprotein E and Alzheimer's disease, risk, mechanisms, and targeting strategies. As you know, the APOE gene is the strongest and most prevalent genetic risk factor among the increasing number of genetic risk factors identified for Alzheimer's disease. APOE is expressed in several organs, but the highest expression is in the liver, and it is followed by the brain. It contains 299 residues, and in humans, APOE has three common isoforms, the APOE2, APOE3, and APOE4. It's different in the amino acid positions, in two amino acid positions. So APOE2 has two cysteines in these two positions, APOE3 has a cysteine and an arginine, and E4 has two arginines. Being homozygous or heterozygous for the E4 allele increases the risk of late onset Alzheimer's disease and lowers the age of onset of Alzheimer's disease. Apolipoprotein E is a major protein of the lipoprotein transport system, and the older and most studied role of APOE is in atherosclerosis and dyslipidemia, and it is also involved in Alzheimer's disease. And lipoproteins, as it is obvious from the name, are complexes of amphipathic proteins that have a hydrophobic and hydrophilic side. So are complexes of proteins with lipids, and their role is to carry lipids through the bloodstream or the cerebrospinal fluid. APOE is a molecule with an exceptional structural flexibility. It contains two structural domains, an amino terminal domain and a carboxy terminal domain. And it's a protein that has a tendency to self-associate and bind hydrophobic substances. And due to its exceptional structural flexibility, as I said, it's a molecule very difficult to study by traditional protein structural analysis. So in 1991, it was solved the X-ray structure of the N-terminal domain of APOE4, and it was shown that it forms a 4-helix bundle. It's a protein very rich in alpha helices, And 20 years later, in 2011, the structure of full-length ApoE was solved by NMR, but in order for the structure to be solved, 5-point mutations were introduced in the molecule that helped it to remain in a monomeric state. So up to now, we don't have an X-ray or NMR structure of wild-type full-length ApoE. However, several based on several techniques, we have gained information on the structure of ApoE. It has been shown that it binds lipids between the N-terminal and the carboxyl domain, and separation of the two domains is necessary in order for the protein to get lipidated and form lipoprotein particles. Furthermore, it has been shown that structural differences between ApoE3 and ApoE4 are located in regions that define both lipid binding and specificity and define the function of ApoE isoforms. In more detail, it has been proposed that this amino acid at this position, which in ApoE4 is an arginine and in ApoE3 is a cysteine, induces different interactions between the amino terminal and the carboxy terminal domain for ApoE4 and ApoE3. And these different interactions are accompanied by different functions for the ApoE isoforms. Furthermore, ApoE is a molecule prone to proteolytic cleavage, and ApoE4 is more labile to proteolysis than ApoE3, and bioactive carboxy terminal truncated fragments of ApoE4, fragments that lack the carboxy terminal domain, have been found in brains of Alzheimer's disease patients and ApoE4 transgenic mice. Furthermore, proteolysis based on animal studies, the proteolysis of ApoE4 is considered to be an early event in the pathogenesis of Alzheimer's disease. In addition, several single amino acid variations have been reported for ApoE, specifically a rare mutation, the leucine 28 proline or ApoE4 Pittsburgh, has been suggested to be a risk factor for Alzheimer's disease, in addition to the risk factor of ApoE4, while recently three more point mutations, that Christchurch, Jacksonville, and this one R251G, have been identified and proposed to be protective for Alzheimer's disease. So these ApoE4 fragments and the rare ApoE variants can be useful tools for gaining insight into the role of ApoE4 in disease pathogenesis. And in our lab, a major activity involves the elucidation of ApoE4 role in Alzheimer's disease by performing structure-function analysis of ApoE variants. A set of studies involved the elucidation of how ApoE4 fragments, specifically carboxyl terminal truncated ApoE4 fragments, how they may affect amyloid beta metabolism. And more specifically, since the APOE4 fragmentation has been suggested to be an early event in the pathogenesis of Alzheimer's disease, we wanted to examine whether these fragments may have an effect on A-beta intraneuronal accumulation, an event that also has been linked to early processes that lead to Alzheimer's disease. For these studies, we expressed in recombinant form two truncated forms of APOE4, one that is called APOE4 185 and lack this region, this section of protein, and a shorter one called APOE4 165 that lacks this domain. And these truncated forms have molecular weights that are within the range of molecular weights of fragments found in brains of Alzheimer's disease patients and APOE4 transgenic mice. And what we observed was that when we treated SK-NSH cells, incubated SK-NSH cells with A-beta 40 and A-beta 42 for 24 hours in the presence of full length and the two truncated forms, we observed that both levels of A-beta 40 and A-beta 42 were greatly reduced in the extracellular medium. And by confocal microscopy, we observed that when cells have been treated with APOE4 165, but not by APOE4 185 or the full-length APOE4, then both A-beta 40 and A-beta 42 was observed inside cells, suggesting that APOE4 165 promotes the cellular uptake of A-beta by this cell line. In order to provide an explanation for this function for APOE4 165, we examine whether it induces any changes in lipid levels of cells. APOE is a lipid binding protein, so it is reasonable to think that it may have such an effect. And indeed, we found that APOE4 165 reduces sphingomyelin levels. And also, for the sake of time, I won't provide details on this technique, but also we found that APOE4 165 also affects cellular membrane fluidity. Changes in membranes composition, lipid composition, and fluidity properties is reasonable to think that that it could mediate passive A-beta diffusion. In addition to the uptake of the identification of A-beta 42 inside cells, we also examined for how long A-beta levels remain in cells, and it seems that A-beta 40 is removed fastly, is catabolized fastly in cells, and after 24 hours we couldn't observe A-beta 40 in cells, but around 50% of the internalized A-beta 42 could be detected in cells after 24 hours. So it seems that internalized A-beta 42 accumulates inside cells. And this A-beta 42 uptake and accumulation increases oxidative stress in cells since we observed an increase in the reactive oxygen species formation. Now, a question we asked is why APOE4-165 promotes the uptake of beta, while APOE4-185 does not have the same effect. So we performed some structural studies, mostly by biophysical techniques, and we observed that APOE4-165 is destabilized compared to APOE4-185, and also it exposes a higher amount of hydrophobic sites in the solvent, and this can mediate more easily initial lipid interactions that then lead to conformational changes, and therefore APOE4-165 seems that can interact more readily with hydrophobic sites of membrane-bound beta and lipids on the cell surface. Next, we wanted to examine whether the allelic state or length of APOE4 fragment, in addition, other lengths in addition to 185, are important factors for intraneuronal beta-42 accumulation. We expressed this 165 fragment also in APOE2 and APOE3 background, and it seems that only APOE4-165 and not the other two alleles is capable to promote the uptake of a beta-42 by cells. Also, we used a more physiologically relevant cell type. We used primary mouse neurons, and we still observe that only APOE4-165 can promote the uptake of a beta-42. We also compared APOE4-165 with 3 other fragments, a longer fragment and 2 shorter fragments. All of them have molecular weights similar to those of fragments found in the brain of patients, and still only this APOE4-165 fragment promotes the uptake of a beta-42 by cells. Subsequently, we examined structural and the conformational differences of APOE4-165 as compared to other allelic states and fragment length. And we found that the E4-165 is thermodynamically destabilized compared to the other 2 alleles, and this destabilization make the molecule more easy to interact with hydrophobic and with membranes and hydrophobic compounds, and therefore we hypothesize this is a way of for APOE4-165 to promote the uptake of a beta by cells. Also we observed differences in salt bridge interactions in APOE3-165 and APOE4-165, and the same applies for the comparison between APOE4-165 and APOE2-165. So it seems that the E4 allele affects the structure and stability of the amino terminal domain of APOE. Comparison of APOE4-165 with the other fragments again showed that this fragment has a unique structure as compared to other length fragments, and overall our study showed that APOE4-165 presents a unique combination of structural integrity and conformational plasticity that may underlie its pathogenic role for Alzheimer's disease. In another set of studies, we focused on this naturally occurring point mutation, the leucine 28 proline, and we wanted to examine what is the molecular basis for increased risk for Alzheimer's disease due to this mutation in APOE4. And again, we performed structural function analysis. The structural studies showed that this mutation induces significant structural and conformational perturbations in APOE4. And although this mutation is in the beginning of the molecule, in the amino terminal site, the beginning of the molecule, it seems that it induces defects that extend to the whole protein, both in the N-terminal and the carbox terminal, and it affects the interactions of the two domains. And we examine whether this mutation may also have an effect on a beta 42 internalization, and we observed that it has the same effect as APOE4-165. It induces the internalization of a beta 42 in SK and SH cells, and it accumulates a part of a beta 42 accumulates in cells for at least 24 hours. And the same happens in mouse primary neurons. And also the mutation induces ROS formation in cells. It seems that the common functional properties of APOE4 L28 proline and the carbox terminal truncated APOE4 165 further support the idea that a beta internalization by neuronal cells is an early event in the pathogenesis of Alzheimer's disease, and this is exacerbated in the case of this point variant. The structural integrity of APOE4 is almost certainly an important component of its role in the pathogenesis of Alzheimer's disease, and the analysis of structurally defective APOE4 variants may hold clues regarding the pathogenic mechanism behind this disease. These studies that I have just presented link E4 proteolysis, which is an early event in Alzheimer's disease pathogenesis, and as well as the effect of this point mutant with intracellular accumulation and oxidative stress that are also early events in pathogenesis of Alzheimer's disease. But we have performed also other studies. The APOE4-185 mutant was shown to induce inflammatory responses in the SK-SH cells. Also, in a research collaborative study, it was shown that APOE4 has reduced binding to complement factor H and this increases A-beta oligomerization and neuroinflammation. We also showed that lipoprotein-associated ApoE4 increases the production of the protein expression of BACE1 in higher levels than ApoE3 and ApoE2, and this increases A-beta production and oligomerization. We have also showed that in the presence of ApoE4, in the presence, of A-beta 42, ApoE4 is prone to oligomerization and leads to neurotoxicity. And current activities involve structural function studies that include also the protective ApoE variants, and we have moved on to study the effect of ApoE fragments and POI mutations in tau phosphorylation and neuroinflammation. The research on Alzheimer's disease has several arms. Definitely one of these is related to ApoE4, and therefore different treatment strategies aim to modulating ApoE function, and one of these strategies aim on the identification of structural correctors of ApoE4. We have performed a series of studies on this, and we use both natural compounds and drug libraries. In a relatively recent study, we identified natural polar phenols that target both the structure and function of ApoE forms related to pathogenic processes involved in Alzheimer's disease. Specifically, we examined the effect of 4 phenols, polyphenols: resveratrol, quercetin, camphorol, and epigallocatechin gallate. We examined the effect of these compounds on intraneuronal A-beta 42 presence in cells that were incubated either with the mutant, POI mutant, or the ApoE4 165 fragment. And what we observed is that when following treatment of cells with the POI mutant and these compounds, resveratrol and camphorol, somehow block the uptake of A-beta 42 by SKNS8 cells. But when the cells were incubated with ApoE4 165, only resveratrol blocked the uptake of A-beta 42. So we examined again whether resveratrol and camphorol may have an effect on the structural properties of our ApoE4 variants, and resveratrol seems to have absolutely no effect on structural properties of either full-length ApoE4, the POI mutant, and the fragment. But camphorol has no effect in the structure of ApoE4 165, a small effect in the structure of ApoE4, but it induces a great, the structure of the POI mutant in the presence of camphorol has great differences. It induces, camphorol seems that it induces structural perturbations, and these perturbations could affect the functional properties of ApoE4 L28 proline. So the effect of of camphorol on point mutation makes the molecule, the protein, to be unable to promote the uptake of beta 42 by cells, while in this case, ApoE4 165 is capable to promote the uptake of beta 42. But why resveratrol blocks the uptake of beta 42? What happens here? One possibility could be that resveratrol somehow does not block the uptake of beta 42, but somehow promotes the catabolism of beta 42 in the cell medium. But we examined this, and it seems that this is not the case. After 24 hours of intubation with resveratrol and ApoE4 in the presence of resveratrol, beta 42 is still intact in the cell medium. Another hypothesis is that resveratrol may affect the membrane fluidity, and by this way blocks the uptake of beta 42. And indeed, we observe that in the presence of resveratrol, we observe changes in the microfluidic state of plasma membrane that may lead to impaired internalization of beta 42 by cells. So we showed for the first time that natural compounds could modify the structure of ApoE4 forms and ameliorate Alzheimer's disease-related pathogenic effects of ApoE4 forms. Just a last slide about our current activities. Currently, we have secured funding to explore the relationship among ApoE and metabolic comorbidities such as dyslipidemia and fatty liver on the pathogenesis and progression of Alzheimer's disease. Metabolic comorbidities seem to affect Alzheimer's disease progression, and since ApoE is a key player in dyslipidemia, then there may be a link among ApoE, dyslipidemia, and Alzheimer's disease. We are currently performing ex vivo studies that examine the effect of ApoE isoforms and variants, protective and detrimental variants in the presence or absence of free fatty acids on various Alzheimer's disease processes. And we perform also in vivo studies. ApoE3 and ApoE4 targeted replacement mice are currently crossed with mouse model of metabolic diseases such as hypertriglyceridemia or fatty liver disease. And then we will examine the effect of these comorbidities on pathogenesis and progression of Alzheimer's disease. These studies are performed in collaboration with my colleague, Professor Dimitris Kardasis from the University of Crete and the Institute of Molecular Biology and Biotechnology. So I would like to thank all my students and colleagues and collaborators in the University of Helsinki, and Boston University, University of Patras, University of Crete, our funding bodies, and of course you for being here today and for your attendance. Thank you.
Thank you, Angeliki. It's really a nice and easy to follow presentations because in the audience there are different, expertise. That's very helpful, so people to understand the molecular basis of and how the mechanism where ApoE4 especially helps the molecular mechanisms of AD starting from amyloid beta. That was a great talk. Thank you. Are there questions in the audience? I have plenty. But if there are from the audience, I will first leave the passage to this, and then I can follow. If
like you, I have at least 10 questions, and I'm going to focus on two. The one is the control, recognizing that there has been a clinical trial of resveratrol in AD, a very robust study, although there have been issues about targeting all comers as opposed to targeting E4 carriers, or better yet, E4 carriers who are prone to snipping into the wrong segments. And for me, which is really critical as a trialist, what's the right dose of resveratrol in humans? So I just was curious if you have any comment about that. My second question isn't really a question, is an encouragement to move these exact experiments into human models that are derived from stem cells. We have cured millions of Alzheimer's mice of Alzheimer's disease, and we're really not showing how that translates into people. So this is wonderful work, but I think you should be doing it in neurons derived from individuals' IPS.
Because that's exactly the point of HIAAD, to bring people together with different expertise. And I'm sure Angelika, who's a great researcher, will take this opportunity. That's why we're here. Angelika?
Yes, this is something we have definitely thought about. Maybe Vasiliki could help us with
exactly.
With that kind of experiments. Regarding resveratrol, yes. Well, I think our study is more of proof of concept. Maybe, well, definitely, someone needs to find, to modify probably the molecule and find a derivative that may be more appropriate in terms of, I mean, we should use lower amounts and a derivative that maybe could cross the blood barrier brain. So a lot more must be done on this.
Well, I would even suggest that you that resveratrol needs to be targeted via biomarker to the genetic subgroup that fits what you have shown. So, for example, giving resveratrol to E3 people, which is about half the AD population, wouldn't be expected to work. And then if you look at folks who are heterohomozygous, perhaps there's a biomarker methodology that could help target and say, these are the E4 people who are more inclined to have the inappropriate proteolysis, and those are the people who should receive resveratrol. In other words, real precision medicine, but the target and the dose become critical parts.
Definitely, yeah.
Yeah, that's a very important point. Is there any... Yes, Vaso, go on, please.
my very good friend and collaborator, a great scientist, Savas Christoforidis, who is here actually in the talk, the presentation, and because he has also IPS, human IPS lines. Hi, Savas. From Yanina, the director of the institute in Yanina, and he has already IPS lines and cells, only because as I said, he's a very close collaborator friend, great scientist, and he's in Greece. So maybe you can start.
It's closer.
Easier.
That's important. Yes.
Yes.
Definitely. Yes.
But in any way, yes, I can help us.
Okay, great. I will communicate. Yes. Thank you.
Okay. Another question, Angeliki, has to do with, since amyloid beta is internalized, would you think that tau will be a nice connection to show if this is affected with tau? And the reason I'm asking is, as you know very well, there is the last 2 years, there's a lot of work with Apolipoprotein and tau pathology, connecting again, ApoE metabolism in the intercellular process and then accumulation of tau. So have you done any of the tau experiments? And that's the first part. And the second is that you think that your mechanism could also explain and be a very nice, connector between amyloid beta and tau pathology, intraneuronal, because I think that's, as you know, this is a kind of a mystery in the basic, or let's say translational biology, the way we see both mechanisms happening in ex vivo or in vitro, but in reality, many times amyloid beta starts in different areas from AD brain and different in tau. So what is your comment on that?
Yes, we have done experiments with tau. We didn't. We observed that, of course, ApoE4 promotes phosphorylation of tau and tau secretion. But now the two forms that we have studied, the point mutant and the fragment, have different functions. I mean, they are not the same now in our set of analysis. Also, we have examined their effect on microglia inflammation. This is an undergoing project, so that's why I didn't present data on this, because we're trying now to elucidate the mechanism behind our findings. But it seems it is discussed also in publications that depending on the cell type and the procedure, the process, ApoE may have different roles. And this is what we see now with the new experiments. Well, the idea was this. I mean, what you said, that ApoE, a specific variant, has an effect on a beta and beta 42, and then has the same effect on tau phosphorylation or neuroinflammation, but it doesn't seem to be a clear association.
Okay, that's good to know. I didn't know that. That's good. Okay. Related with the liver, as you refer, how would you see the connection? Because I know some work from HIAAD and Wilson Lab in Stockholm, for example, working on liver and ApoE, not so much in Alzheimer, but in general, in the context of Alzheimer, but it has to do with the humanized liver mice, where they see cognitive deficits when they are expressing ApoE4, and as well as when they express other studies when they express just amyloid beta locally in the liver. How would you think the connection with the liver metabolism, including ApoE or excluding ApoE, in the precipitation of AD, at least in the mechanism?
It seems, well, there are studies that have shown that ApoE in the periphery has an effect on, it induces the development and progression of Alzheimer's disease. They have done, I don't have the slide right now, they have done some elaborate experiments. I think there was no expression of ApoE3 or ApoE4 in the brain in a model. It was only in the liver and the periphery. And the ApoE4 mice developed Alzheimer's disease and not the ApoE3.
Yes. Yes. Any more questions from the audience, guys? I don't see anything here in the chat. But if we don't have more questions, we would like to thank again Dr. Chroni, Angelika, with a great caller. I'm happy if these seminars, is a bridge, as we exactly said in the title, to start new collaborations inside and outside Greece, because the country is not very important, the science is important, and that's why this seminar trying to bring together basic, translational, and clinical researchers. So that will be a great opportunity, not only for changing and putting together efforts, but also for brand applications in future. Once more, thanks again for being with us.
Thank you very much.
Dr. Chroni. Thanks for the audience, and we will have a summer break in August. We will be back in September with more seminars happening. We are promising to have a lot of nice surprises, new ideas, and out of the box ideas. Like today, which we knew a bit, but now we have a little, more clear view of how this could contribute to the precipitation to the pathology. Thank you very much for all your participation in this seminar, and thanks for the audience to be with us. Have a nice summer.